Welcome back, students. This is video lesson 2 in continuation with video lesson first based on chapter 2 sexual reproduction in flowering plants. In video lesson 1 we discussed about the structure of stamen. There we studied that stamen is divided into two parts, anther and filament, where anther is the fertile part of the stamen. Now, there are some technical terms that we are going to study today related to anther. Generally, anther is bilobed. What does it mean? It means that each anther has two lobes. Bilobed anther is diathecous. What does it mean? It means that each lobe has two chambers or two cavities. And bilobed diathecous anther is tetrasporangiate. Tetra means 4. So here we can see that diathecous anther has 4 microsporangia, or you can say that bilobed anther has 4 microsporangia. What does it mean? It means that each lobe has 2 microsporangia, which can be easily visible when we take transverse section of anther. So each lobe has 2 microsporangia. So in total an anther has 4 microsporangia. That is why anther is called tetrasporangiate. So these are the technical terms related to anther. There are some exceptional cases seen. For example, Wolffia, where we find only 1 microsporangium per anther lobe. Now students, let us discuss about different wall layers of anther and their functions, where the microspores are produced and how are they produced. Okay, now wall layers can be easily studied when we take the transverse section of an anther. This is a transverse section of young anther. So here we can see that there are 4 microsporangia. Each microsporangia are separated from each other. You can say these are the 2 microsporangia which are separated from outside by a shallow groove, and internally you can see that there is a parenchymatous strip which separates the 2 microsporangia. Now you can see here that the 2 lobes, this is one lobe, this is another lobe, are separated anteriorly, but at the back side the 2 lobes get connected by a connective, which is a sterile parenchymatous tissue. Here in the connective is present a vascular strand which is meant to supply water, minerals and food to the anther. Now here we can see that each microsporangium is surrounded by different wall layers. So let us study any one microsporangium and its wall layers. Why we are going to study only one microsporangium? Why we are supposed to label only one microsporangium? Because whatever the wall layers shared by one microsporangium, rest of the microsporangia share same wall layers. Now the very outermost layer of microsporangia is epidermis. This is well defined epidermis, common wall of anther. Now next is endothecium. Endothecium is also called fibrous layer because it has deposition of alpha cellulose fibrous thickenings. That is why endothecium is also called fibrous layers. Now next is middle layers, which is 1 to 3 layers thick. Now at maturity middle layer cells shrivel off. They you can say wither off. Now here next to the middle layers you find a layer which covers the internal tissue, that is called tapetum. Now here we can see tapetum is of 2 types. One is amoeboidic, another is secretory. Tapetum cells are very special. They are generally multinucleated. they have full of cytoplasm and nutrients. Now here we can see that in certain cases the nuclei of tapetum may become polyploid due to endopolyploidy. You can say. Now here we can see that tapetum cells are of two types. One is amoeboidic, which is invasive, and another is secretory. Now if we talk about the invasive type of tapetum, they passes into the microsporogenous tissue and nourish them. They provide nutrients to the developing microspore mother cells. While if we talk about secretory tapetum cells, they secrete some substances which help in growth and differentiation of pollen grains. Now here we can see that at the center of the microsporangium lies sporogenous tissue, which is the fertile tissue. All the cells of sporogenous tissue directly behave as microspore mother cells, which are diploid in nature. Now here you can see that all the microspore mother cells are separated from each other by forming a callose cell wall. Deposition of callose around pollen mother cells cell wall. So each microspore mother cell gets separated from each other and they become round and they undergoes meiosis. And after meiosis, from one microspore mother cells, 4 microspore are produced. So here you can say that from one microspore mother cell, 4 microspores are produced. So these microspores, or you can say these pollen grains, are arranged in the form of tetrad. So the arrangement of microspores in a tetrad may be you can say isobilateral, which is most common type. It may be tetrahedral, it may be T-shaped, it may be decussate, it may be you can say linear. It depends on species to species, but most common type of pollen grain arrangement in a tetrad is isobilateral. But in Aristolochia elegans, we can see that all the 5 types of pollen tetrads are visible. Now here we can see that tapetum cells have some functions, some special functions. What are the functions of tapetum cells? Let us recall. Number 1, that tapetal cells nourish the developing pollen grains. Number 1. Number 2, tapetum cells secrete some special enzyme, say callase enzyme, which degrade callose around the pollen grains so that pollen grains get separated from each other. Another function of tapetum is that they produce indole acetic acid, which is meant for germination of, or you can say elongation of pollen tube or growth of pollen tube. Now tapetum cells also secrete some lipid rich Huebsch granules, which is meant for formation of sporopollenin around exine of the pollen grains. So we will discuss what is sporopollenin later on. Now tapetum cells also help in formation of pollen kit, which is oily sticky substance rich in you can say lipids and carotenoids, and this pollen kit is very essential for pollination by insects. So in this way tapetum helps. Now tapetum cells in some cases also secrete some special proteins which help pollen grains to recognize compatibility with stigma. Now here we can see that the main generative tissue or sporogenous tissue are responsible for formation of pollen grains, as I told you earlier. Now here we can see that all the sporogenous tissue directly behave as microspore mother cells. Microspore mother cells are diploid. They undergo meiosis, meiosis 1 and meiosis 2. After meiosis 1, microspore diodes are formed, and after meiosis II, microspore tetrads are formed. Now after formation of microspores, or you can say after formation of pollen grains, we can say that sporogenesis resulted. More specifically, microsporogenesis resulted. What is microsporogenesis? Formation of microspores from microspore mother cells by the process of meiosis. What is the site of microsporogenesis? Anther. More specifically, microsporangia. You can see here, these are the sites where pollen grains are produced, and pollen grains are first cell of male gametophyte. So students, in my next video I am going to tell about the development of male gametophyte in angiosperm. Thank you and have a nice day.